to you. All right, Ethan, this is for you. Why does it say I'm muted? All right, Ethan, we're making this video for you. This is how you're going to assess how you're going to go over your answers on the last test.
Yeah.
Let's get that far enough away. I should have sent you your response on the day we did it in class, I think.
I literally remember seeing it.
Okay.
So I know I have.
Yeah, it's in there somewhere. If your email is like mine right now, I just wish people would stop emailing me. I can't find anything. It's like, yo, y'all gotta stop. And then all the Google Classroom stuff that hits automatically. And I do like 3 weeks. I do 3 weeks at a time when I enter those.
Oh, okay. Yeah.
So when you get your bio assignments, it's like, all right, so Ethan, you're gonna open yours. And I'm gonna share my screen. And we'll get started. All right, cool. So you can see your answers. All right, and then hopefully this works. This is, I hope this is visible enough, Ethan. But let's give it a shot. All right, so question 1. What order did you put these in? So what did you have?
It says someone else. It says someone else. This is Miriam's.
You have Miriam's test?
I think so.
Huh.
Yeah.
Why would it send? How could it email you hers?
I'm not sure.
Because she has a different email. Okay, let me.
The updates and yeah, I'll be in touch with
All right, I'm going to release score to
whatever else we
you.
need to do.
And we'll do Ethan's also. Okay, this new email should have yours, actually.
Sorry.
That's okay. Yeah, I'm sorry. I just,
Nope.
Yeah.
You're totally good.
Thanks. I appreciate it.
All right. See ya.
Do you need your weird glasses things?
Those are not mine.
Oh.
Those are.
Huh.
All right, toodles.
See you. Got it?
Okay.
Nice. All right, so this is what we did in class that y'all both missed. Actually, 5 people missed this class. Yeah. So what order did you put these in?
D, E, A, C, E, F.
So what did this look like to you?
That was wait, like stage
Yeah.
fertilization.
Yeah, fertilized egg. You might have thought of it as a zygote or embryonic stem cell. All those would be correct.
Yeah.
What does this look like to you?
Early cleavages.
Sure, love it. Yeah, absolutely. This could have been early cleavages. You could even say maybe early blastula.
Okay. Yeah.
Maybe, right?
Right.
If this is a forming blastocoel.
Right.
I called this early cleavages too. And then you said A, you said?
Yeah, gastrulation.
Yeah, this is almost certainly gastrulation. There's lots of cells in here. Things are going in, things are swirling around.
Yeah.
See, you said?
I think I said organogenesis, like.
Yeah, absolutely. This is early organogenesis. So like this is like the 24-hour slides. It's not in the fetal position yet. We're looking down on it.
Right.
There aren't structures are forming, but there aren't real organs yet. And then this one?
That's just further along organogenesis, right?
Yep. What's one thing you can identify in here that makes you go like, yeah, further along?
It looks like it's in the fetal position.
Yeah, fetal position. What about structure-wise?
It's hard to like depict. Cause like even F, like it took me forever to find out that was like a bird head.
Yeah, sorry about that.
I had to look
Sorry
at it,
about
like,
that.
3 different
What'd you call
angles.
this one?
I think I just said like fully developed.
Yeah,
bird.
sure. Yeah, or
Yeah.
hatchling or at hatch.
Yeah.
Well, I would say this is actually an eye, sort of, right?
Okay, yeah.
The eye placode, and you've
Maybe a
got like,
heart.
yeah, the heart. Boom. Cool. All right. So the heart is formed from what kind of, you know what? I'm going to actually repeat your answers because I bet he can't hear them.
Okay.
Yeah, so this is later in organogenesis, and you said this is a heart, and we said this is maybe an eye placode. What germ layers make the heart?
Ooh, endoderm.
Inside layer?
Oh wait, mesoderm.
Yeah, mesoderm, right? So this is a mesoderm derivative right here. That's cool. That comes up later. That's a question. Okay. Yeah, so well done. Those are all correct. One of the problems with the software, right, is I tried to start putting keys in, but literally if you did yours on lowercase, you separate them by commas. Like if you do anything slightly differently, you can answer the correct answer, but it doesn't give you credit.
Yeah, do comments.
Yeah. So the software doesn't grade this stuff, but it actually gives us an opportunity to correct any misconceptions before we move on, and then learn the stuff and then earn for full credit. That's the plan.
Wait, so should I be doing
No,
checking the software?
no, you're just keeping track for your own head, like what you got right, and what you didn't get right. All right, so stage of development for A, you already said was gastrulation. Stage of development for B, you already said was organogenesis. The stage of development for letter C, you said was early organogenesis or organogenesis. Letter D, you said fertilization, fertilized egg. You said cleavage or early cleavage for E. You said fully developed bird or ice. Cool. And I said hatch. Same, those are correct. This is a tricky one. Okay. If each of these represents a vertebrate, but you don't know what kind, what are points in time when you know for sure the Spiemon organizer is active?
I was very confused by this one.
Okay,
This
so
was like
let me give you an example. So in the frog, fertilization activates the Spiemon organizer gene expression on the opposite side. But in a mammal, the Spiemon organizer doesn't start to activate until 16 cell stage.
Okay.
So in other words, that's the latest we've identified Spiemon organizer those genes starting to get expressed and cause things to happen.
Right.
And we know the Spiemon organizer forms the dorsal lip of the plastic. Gastrulation's happening definitely. So what did you put on this one?
I put, this was like 75% of guess. I put A and C.
Nice. A and C? Yeah. Absolutely gastrulation.
I'm still
absolutely when neural tube is folding, because this is the beginning of organogenesis.
Mhm.
Now, interestingly, the same genes that run the organizer also run what's called the new COP center, new COOP, and NIEUWCOOP, somebody's name, some maybe Dutch or something, right? Like very complicated. A scientist. But it's the same genes doing that organizing center that builds the brain from anterior to posterior as it is the organizer. So I'm just calling it the same thing. So you could have said B. I feel most comfortably A and C, you know for sure. E's kind of you guessing if this is cross section, is this 16 cells?
Yeah.
So I think it's acceptable to say E as well, but I understand people not saying it. You didn't say F, did you?
No.
Right. So the organizer's done. It's not
exactly.
doing anything anymore, right?
And D, it hasn't
Exactly. Only in a frog does the organizer get activated this way. Right. Nothing else, right? Everything else is later. Or sorry, salamander. Well, salamander and a frog. Amphibians. Okay, probably all amphibians. Cool, groovy. All right. So this one is the name of each structure. So this is the crazy long one. Okay.
I used the diagram for this one.
All right. What'd you say for number one?
Hindlimb bud.
Ah, nice. That's what I intended. What if someone said somites.
Okay, yeah, I had that label
Yeah.
on one of the.
Well, you buy it, right? Because
Yeah.
these chunky things that grow from anterior to posterior are actually somites.
Yeah.
Even though this is what I intended, somites would have been sensible.
What are their
Sensible.
functions?
Somites are balls of mesoderm that form. It's like if I cut this embryo right through here, right, and I look at a cross section where this side is dorsal, right, dorsal, and this side's ventral. Okay. What I've just done is I've got the primordial brain is here. The notochord is here. The mouth, the anterior intestinal portal is forming here. So that's going to be the endoderm tube that runs all the way through the organism. It's actually, at this point, it's actually like this. It's open to the yolk. Okay. And on either side of the notochord, these balls of mesoderm form, these chunks. And they form these primordial tissues called myotome and sclerotome, which I didn't teach you this year. The sclerotome forms largely from signaling from the ectoderm, and the myotome forms because of genes expressed by cells around the notochord. And this forms bone on the bottom, the sclerotome, and on top, the myotome forms muscle. And so this bone is going to wrap around the notochord and form, that's dorsal, and basically form your vertebrae. Now we're looking
That's
at
what your vertebrae is basically made out of.
Exactly. It's the primordial tissue that forms vertebrae, the somites, and ribs and other skeletal structures moving down. And the myotome forms all the body muscle wall associated with those bones. So it builds your skeletal and muscular system as the embryo moves down. But in this stage, there's no muscles yet or skeleton. Even cartilage hasn't formed yet. And we're going to talk about how bones formed in our very last lab, which is it's an amazing process. And it's the reason why when you break a bone, it heals like that. But if you break like soft tissue, it isn't hard bone, it takes forever and it doesn't heal right, right? So it's just these balls of tissue that form your skeleton and muscle as development proceeds. Cotton.
I forgot.
Yeah. It's bad. What'd you call this thing?
2. I did not label 2.
Yeah, that's nothing.
Oh, okay, good.
Yeah. You're the only person that didn't label it.
Really?
Everybody else, So I said it's extra embryonic tissue, which means it's not part of the embryo. That's all I said, but I wanted to sound smart. A lot of people call this a vitelline vein or artery. It could be, right?
Oh,
that makes the most sense.
I have that for 3.
Yep. This is definitely a vein or artery. These are veins or arteries, or you could call them vessels. Vessels. And vitelline is what we call these before all the structures have formed as well, right? Number 4.
AER forelimb bud.
Nice. That's what I intended.
This is going well.
Yeah. But now what's interesting is could you imagine someone calling this potentially the forelimb bud?
Why there?
Because it's kind of in the rightish place.
Yeah, general area.
Yeah. So I could imagine somebody calling this a somite because it's still right next to the somites, right? This is also right next to the somites. You might even call this notochord. What'd you call it?
Yeah, I said notochord.
Dang. That's what I intended.
Wow.
You did a lot better than you thought you did.
Yeah.
Oh my God. Okay, so yeah, this is the notochord, but again, you could have called this somites.
And notochord vesicle, that's what I had labeled in the diagram. Is that right?
I don't know what a notochord vesicle is.
Oh,
That's a
maybe
little-
I had it like 2 things labeled and they were like right next to each other, so I thought it was notochord vesicle.
Yeah, probably. You might have, except for, because this is called the otic capsule or vesicle.
Okay. And
It's
there was
not
those
numbered in
two.
this thing, But I imagine if I wrote it in a drawing, it might have been right behind it. It might have looked like it said Notochord vesicle.
Yeah, that's
Yeah.
probably-
That makes sense. What'd you call number 6?
I did not have anything for 6.
So there's a lot of things this could be.
Okay.
But it does look like it's not part of the embryo. What'd
Yeah.
you call 7?
7, nasal artery.
Nasal artery. Huh. That's weird.
Oh, is that?
Okay, what'd
It
you
looks
call
like-
what'd you call 10 then?
10 is telecephalon.
Telencephalon. So this is the forebrain then.
Yeah.
Okay,
10,
so
14.
now here's the interesting thing. Hold on.
Yeah.
Or forebrain. So I intended this to be the telencephalon.
Okay.
Now hold on. However, if you labeled this the telencephalon,
Yeah.
then you must have labeled this what?
The
Diencephalon, which is the midbrain. Great. And then that means you labeled that
the other one.
the mesencephalon. Yeah.
Yeah.
Great. So all of your answers are consistent. Diencephalon, which means you know what you're doing. You know your way around the embryo. I would call this, your answer is correct. Even though I intended it to go like this. I intended telencephalon 7, diencephalon 10, mesencephalon 14, but then you would have had to call 12 the rhombomeres. But it doesn't matter. It's irrelevant, right? If you had skipped the hindbrain and called this rhombomeres, then it's like you don't know what you're doing. So the way you labeled it correctly. If you labeled this something, you could have called it the anterior intestinal portal, like I'm trying to point to the mouth around the other side of the head. Or you could have just said mouth, but that's not fancy.
It's right next to the eye.
And this could also be, I intended this to be possibly the nasal pit, or this could be the nose. But this is like you're labeling this line going to the nose, so nasal artery also makes sense.
Okay.
What'd you call 9?
Eye.
Nice. Yeah. Eye placode, eye vesicle, all those are correct. Okay, 13. Somites? See, you need somites somewhere.
Right.
I think I intended on people identifying that as somites. What'd you call 8?
Heart.
Nice, that's the heart. And 11?
Bronchial artery.
Yeah, bronchial arches, actually.
Oh, artery.
Well, they form the bronchial arteries. So it's, again, also correct.
Right. It's just like the stage of development it's in. It's
Precisely.
okay.
Yeah, things are changing names. And so because these things are fluid, you can't actually decide only one thing is correct. Because it depends on, and I've not labeled the hours.
Right.
Exactly. So it'd be the same thing as if you labeled, if you labeled 7, 10, and 14 as neural tube.
Yeah.
I have to give you credit because it was the neural tube. It's a little lumpier. I think it's forming brain now, and like you're not wrong. Right. So that's the complexity of questions like this. What stage of development did you call this thing?
I said early organogenesis, and I said 80 hours just because on that document you shared with us, I picked the picture that it looked most similar to,
Uh-huh.
and it looked most similar to the 80-hour one.
Well done. That's what I drew it from. So
Oh wow, perfect.
yeah. So that's why I don't think it looked enough like it that people would recognize it. Yeah. Anything that says, you could also say pharyngula. That's the point at which organs start to form, but they're not vertebrate specific. So all organisms look the same at the pharyngula phase of organogenesis. Okay, so I know what you said for number 10. Based on how you labeled it, you checked 4.
I actually checked 5.
Huh.
I'm not gonna confused.
Yeah, because you labeled 4 the AER.
Yeah,
And this says what number indicates the anterior most expression boundary of HOC6?
yeah, and the AER.
Yeah,
I think
so
it's-
we have to remember that HOX6 is expressed at the anterior-most boundary of, sorry, the limb bud grows at the anterior-most boundary of HOX6 expression. Yeah, groovy. So 11, you said,
AR.
Nice. And then you said,
For the two genes, FGF4 and Sonic.
great, yeah, so 11's apical ectodermal ridge or AR. You could have said forelimb bud,
Yeah.
actually, now that we know that those two things are synonymous, or limb bud. And then 12 would be sonic hedgehog or FGF4. Anterior-most region of the embryo, what'd you say?
I checked 2. I checked 7 and 6.
Huh, that's what I would have done.
Oh, okay.
Yeah. It's 7 or 6. Now that it's in the fetal position, right? This isn't up here. 13 is not anterior anymore.
Right.
Yeah. So this is,
So the most posterior would be.
yep. Absolutely. Whoops. What just happened? Okay. Blah, 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 blah. What genes are expressed in the region labeled with the number 9?
Oh yeah, these ones I didn't understand. I just got these from the notes. OTX1, OTX2.
Yeah. I mean, names of genes don't have anything in them to understand.
Yeah.
Yeah. So you didn't say any others?
No, that was it.
Okay. So that's correct.
Okay.
So what happens is, whoops. OTX1 and OTX2 are expressed here. And they're expressed as cells grow out in a tube that connects to here. And then PAX2 and PAX6 are expressed on the outside of the ectoderm, and all together, OTX1 and OTX2 and PAX2 and PAX6 build the eye from both sides. So all of those are correct answers.
I actually had for the genes are expressed in number 7. I have PAX2 and PAX6.
Yep. That's also correct.
Oh, okay.
They're overlapping expression boundaries.
Okay, good.
Okay,
it's
so
not just the eye. That it,
well, I mean, no, it's also the overlying ectoderm, and since this is a transparent embryo, and the overlying ectoderm is expressing PAX2 and PAX6 in the most anterior region, I can't say that. I mean, PAX2 and PAX6 is expressed where 7 is expressed, or where 7 is, because you can. I didn't say on the outside or inside.
Yeah.
I could have tried to be more specific, but then that is somewhat confusing, right? And we gotta see this thing as 3-dimensional, and gene expression boundaries is overlapping. So PAX2, PAX6. What else did you say?
Snail gene.
Yeah. Yep, the snail gene is the one that is being expressed. Again, if we do this in cross section, you've got your neural tube, and here, snails making cells migrate out here in this anterior-most. Okay, what else did you say?
I think that's it. Yeah.
You're supposed to say 5.
Yeah,
So
I
you
got
could
it.
say
That's it.
follistatin, beta-catenin,
I think I had these ones down.
CMOs. These are the what the organizer genes that start to build the brain are called from front to back. Follistatin, beta-catenin, CMOA. And then as we go further back, you've got OTX1 and OTX2, right? OTX2, you've got DLX. God, what other genes are expressed here? Did I write something down? I'm tired and old. Let me check. Oh yeah, cordon. Of course, cordon and noggin. Your head genes, actually named after a head, right? And this is like a cord, a spinal cord, neural cord, cordon, noggin. Better than like PAX6 or OTX, right? At least those are named after what they make, right? And snail, why?
It's so rare.
Yeah, because the cells migrate slowly. That they, I don't know, like, and then Wnt1 is the other. Sorry, that causes neural crest to migrate. Right. Remember, these are the genes that in the anteriormost neural crest didn't get expressed. So the cells just stayed stuck there and they didn't go around and build a face in Quail Johnson, right? Crazy town. Can't believe that actually happened. I really got to take that embryo with me. It's a 1 in a million chance. Like, yeah. We already said the heart formed from mesoderm. What'd you say for 14? Your brain.
OTX1 for number 9.
No, number 17.
Oh, number 17.
Yeah, we made it to the end, I think. Ectoderm, yeah. Boom. That's the test. Well done.
Wow, so I was a lot less.
Yeah. So